A todos e a todas.
Good morning, everyone. I am Gustavo Rafael Rodobossetti, Research and Development Manager of the Sanepar Company, Coordinator of the Thematic Group of Wastewater Treatment and Brazilian Association of Sanitation Engineering and ABES-PR, and we are transmitting at this time in Portuguese and English. So if you feel more comfortable in the company watching this event in either of those two languages, please access the links that are available on the chat. It's with great pleasure that we begin the third special session for Brazil Water Week, the most important international event on water held in our country in Brazil. Brazil Water Week will be held between the days the 23rd and 27th of May in an exclusive platform and interactive platform. And the website for registration is on the chat as well, and you can just connect to it. Gustavo, here in this channel of ABES Sanitation, you participate in these special sessions free of charge. They're already occurring and will occur over the next few years and next few days rather until Friday. And I would like to briefly highlight that because Friday the 13th, we had a special webinar talking specifically about epidemiological surveillance based on wastewater monitoring. Here, some experience in the state of Paraná for facing COVID-19. So we've had important collaborations through monitoring done in Curitiba and in Foz do Iguaçu, and important leadership were with us, important researchers. So if you didn't watch this event, access the link that is in the chat and watch. It's free of charge, and there's a lot of important information to be shared. Obviously, at that time, we were talking about experiences that would happen essentially in the state of Paraná in Brazil. But today, we have a special session that will also deal with this same subject. So we will talk about wastewater monitoring as a epidemiological surveillance tool for COVID-19 and bringing experiences from around the world. Knowledge that was produced over the last year and a half or 2 years that are in service of the population. To be able to talk a little more about these global experiences that are very aligned with the proposal of Brazil Water Week, we have very illustrious guests, professionals with enormous knowledge in the field and professionals that have been coordinating and accompanying step by step the application of epidemiology based on wastewater surveillance for COVID-19 and facing COVID-19 or other possible applications that can be developed from the use of this technique. And in this sense, we will have the opportunity to hear about the experiences in the United States. And to that end, we will have the help of Angela Harris, professor of the North Carolina State University. Angela, welcome indeed. And then next, we will have the opportunity to listen to Hubert, Chief of the monitoring sector, environmental monitoring sector of the Health Security Agency of the UK. Hubert will give us the experiences and developments in the UK that will have been produced very important results which have been published as well. And we will finally consolidate the technical presentations with Professor César Mota of the Federal University of Minas Gerais. He is the coordinator of the National Institute of Science and Technology in Sustainable Wastewater Treatment Plants, and also the COVID monitoring network for wastewater, which is done in partnership with the National Water and Sanitation Agency, ANA. After the presentations then of the experiences of the United States, the UK, and the Brazilian network, which today monitors important capital cities, state capitals in our country, we will have a debate and this debate will be moderated by a professional Thais Araújo, Cavendish. Thais is a general coordinator of health and environmental monitoring of the Ministry of Health in Brazil. So Thais will moderate the discussions and provide questions and reflections for our illustrious participants. So I thank you all for being available and willing to join us. Angela, Hubert, and Thais, and César, and I thank you all for those who are watching us and who will certainly collaborate throughout this journey. You are all very welcome indeed to this webinar, and more than that, thank you very much, everybody, for accepting the invitation, allowing us to be at this moment dialoguing with all of you. So my friends, to know a little bit more about each of our guests, just access the tab under the video. You will have a little more information about the CV of each guest. It's very important and interesting to know more about these guests and their scientific production, especially in this area of wastewater monitoring. And to those who are watching, who have the opportunity to participate actively in our event. To do that, you just need to put in your comments or questions or reflections in the chat below. We have a team of IT professionals of ABES are dedicated to answer or to at least read all of these questions and comments, and all of these will be forwarded to our guests at the end of the presentation. So let us move on to the technical content of our important event. And to that end, I pass the floor at this moment to Dr. Angela Harris, who will bring the experiences of the United States. Angela, once again, thank you very much for being here with us. It will be an honor to hear you, my dear friend. The floor is yours.
Great, thanks so much for that introduction.
Obrigada por essa apresentação.
So I'm hearing, okay, so thanks so much for that introduction. And I'm very honored to have received this invitation to speak with you all today. I'm a professor in civil construction and environmental engineering at North Carolina State University. And I've been involved in some wastewater-based surveillance efforts here in the US, particularly for the state of North Carolina, but it's been a part of a national surveillance program. And I'm excited to share some of the insights that we've gleaned since the pandemic started, and also discuss some future directions for wastewater-based surveillance and some broader applications. So disease surveillance, I just wanted to quickly start about why is disease surveillance important for public health efforts? Disease surveillance can really be important for determining how we allocate our resources, understanding where and to what extent disease is present in the population is really important whenever we think about how to address those issues. It's also important for different management decisions that might be made, such as stay-at-home orders or mask mandates or testing locations and accessibility. But also for identifying potential sources or situations where disease has been spreading. And so disease surveillance can be important for these many different applications and from different stakeholders. But when we think about disease surveillance, the main goal is to identify, all the infections, symptomatic and asymptomatic infected individuals in a population. That would be kind of the ideal metric that we could use. However, we're often not able to know that exact number. And so sometimes we would then look at symptomatic individuals that are tested, because often people won't go get tested unless they are symptomatic. But then some of the common health metrics are often cases that are actually reported. And so this would mean that you would have to have some sort of system for reporting of cases. Hospitalizations or intensive care occupancy are also common metrics for disease surveillance, because these can be recorded in hospitals and put into databases that are more easily accessible and monitored. And similarly, deaths are also a part of a common disease surveillance metric that public health officials often use. I want to kind of go back to this lower portion of of this pyramid in terms of what are we trying to target in disease surveillance efforts. And this is where wastewater-based surveillance can play a nice role. If the pathogen is shed in the feces of infected individuals, then this could offer an opportunity for us to monitor all of the infections in a population, whether individuals are symptomatic or asymptomatic, and doesn't rely on individualized testing. And this is where we can use our sewer systems. And so once again, the population that would be surveyed would need to be connected to a sewage system. So this might not work in more rural areas that have on-site systems, but in urban areas or in more densely populated areas that are connected to a sewage system, it can actually composite the waste, the fecal material from an entire population that we can then gain insight on symptomatic and asymptomatic infections in that population. Wastewater-based surveillance has been used previously prior to the COVID-19 pandemic. The influenza, the flu has been monitored in wastewater, polio virus, and also opioids. So illicit drug use has also been monitored using wastewater. But I would argue that really wastewater surveillance has taken off since the COVID-19 pandemic, given a lot of the challenges and the need for data on prevalence in the population. A lot of investment has been made in wastewater-based surveillance, particularly in the US. And the reason was that pretty early on, we found that some researchers, so this is data from a study conducted in China, that SARS-CoV-2, the virus that causes COVID-19 disease, is the RNA of the virus is shed in feces. And there are even cases, so in yellow, these are that the fecal sample is positive, and then in red, this is where the throat sample was positive. And this is showing kind of over time going along the x-axis, and this is for 41 different patients. And it identified that even there were instances where the feces were still positive for this RNA of the virus, where the throat, the swabs were not positive, and so that you weren't detecting it in the throat swab samples. And so this just highlighted, the potential for using feces to monitor prevalence in the population. I will note that this is also a limitation, because it could be the case that someone is shedding the RNA in their feces, but are no longer contagious and able to spread the virus. And so there are some limitations with this, but given that it's found in feces, it really highlighted its potential at using wastewater as a surveillance tool. And so early on, there were some studies that looked at the relationship between clinical cases in a population and the concentration of the viral targets of SARS-CoV-2 in wastewater. And what was observed was that there was a correlation between the two, and that wastewater was actually a leading indicator of the clinical cases. And from some of these early studies, it really pushed the expansion of these efforts. Yes, sorry. So when I was started to be involved in this work, which it was, very early in 2020, once it was becoming obvious that SARS-CoV-2 would be spreading globally, we received some funding to explore using wastewater as a surveillance tool from the National Science Foundation. And some of the key questions that we needed to answer was, first, how do we measure virus presence in wastewater? I have done a lot of environmental monitoring of pathogens, and every media is unique and every pathogen is unique. And so even though some of the steps would be quite similar, you had to optimize them in order to be able to have a valid method and a reliable method. Then we were really wanting to look at the trends that we see in wastewater compared to conventional metrics that public health officials use. And then we also asked the question on how should we be reporting these trends so that public health officials can make decisions, because it's not just having the data, but how the data are presented would be really important for making sure the data are used and are helpful. So my work has been in the state of North Carolina, and one of the systems that we first investigated was the Raleigh sewer shed. And this captures wastewater from about 580,000 residents. And we actually had 5 different sampling points in the Raleigh sewer shed. We did at the head of the treatment plant, where we sampled both influent and primary solids that were settled out of the clarifiers. But we also did some sub-sewer shed monitoring at some neighborhoods and downstream of a hospital. But for today's presentation, I'm just going to be focusing on at the head of the plant location. So we wanted to compare using influent, so the liquid wastewater raw influent, to using these kind of settled solids, some of the primary solids that have already been settled out. And the reason why we wanted to compare this was because solids are already somewhat concentrated. You won't necessarily have to do a concentration step as you would with influent. And others had shown that you're more likely, it's a more sensitive matrix, that you're more likely to have detection in solids as compared to influent. However, solids may be more inhibited. There would be a lot of other material that's also concentrated in the solids that could impact the detection method. And depending on how your plant is operating, the solids retention time may actually impact your ability to kind of be a leading indicator of the clinical cases. I don't want to go into detail about our methods, but I just want to say that we, for the influent, we use a concentration step using vacuum membrane filtration. And for the solids, we just did a direct extraction of the nucleic acid from those solids. And then we use digital droplet PCR, where the system breaks your sample, the nucleic acid extract into 20,000 droplets, and a targeted PCR reaction occurs, and then it actually will measure fluorescence in each individual droplet to estimate it's an absolute estimation of the concentration of your gene target in the wastewater. And so we did a lot of different piloting to optimize and to develop a modified RNA extraction protocol, and we now we feel pretty excited about the sensitivity and specificity of our assay. So now I'm showing you the trends between the solids and the influent over time, and so this is data from 2020 March all the way to December. And this is where we are collecting samples 2 to 3 times a week. And this was representing a rolling average of 3 sample events. And so the data have been smooth from this rolling average. So in blue, we have the concentrations of a SARS-CoV-2 gene target N1 in the influent. And then in this kind of brownish gray color is the concentration that are in our solids. And we can see that the concentrations are quite correlated. I will note that this is a pretty strong correlation using a Spearman rank correlation coefficient of it's greater than 0.7. And we do see that the influent does have a slight lead to the solids, but that we are quite pleased to see how correlated these two indicators were. And we had more detections in the solids. Only like 2 of the samples were, all of the samples were detected for the solids, but in the influent, there were 3 samples that had non-detects. But then our next question that the public health officials were really wanting to know about were how do these wastewater metrics compare to indicators that they were already using? So clinically confirmed cases and also COVID-like illness as recorded during emergency room visits. And so these were 2 indicators that are often used for other diseases and that were being used for SARS-CoV-2 infections as well. And so here on the top is that same figure where we're looking over time and we see the influent compared to the solids. And then the figure below is in red, the line is the clinically confirmed cases over time. And this is also it's a 7-day rolling average. And then we have the COVID-like illness down at the bottom in the yellow. And all of these indicators are quite correlated with similarly a Spearman's rank correlation of 0.7 or greater. The 2 indicators that were most closely related were the COVID-like illness and the clinically confirmed cases. However, the correlations were still, that correlation coefficient was 0.84. As compared to 0.7 or 0.75 with the wastewater indicators. But once again, our public health officials that were very closely partnered with this work were very happy to see such a strong correlation, and it really provided a lot of motivation for continued efforts to continue with wastewater surveillance. I do want to just discuss briefly how we were also looking at some of the data in terms of trends. And trends were what we were using, because given that the concentration of the virus in wastewater could change because of many different reasons, because of the variant and the concentration that's shed in feces, sometimes with rainfall, the strength of the wastewater would change because of inflow or infiltration into the system where stormwater is getting into the wastewater sewage lines. But we do look at trends and how whether there were concentrations were increasing or decreasing, so that the health officials would know we need to start being concerned, or we could start considering lifting different mandates or making different management decisions. And so one of the methods for trends comparisons was using a regression model that would use date as the independent variable. And if this independent variable was statistically significant, we would then be able to say that there was a significant increase or a significant decrease in the concentrations over time. So 3 data points were classified as a short-term trend, whereas 7 data points were classified as a long-term trend. And these metrics were developed with the Center for Disease Control, the US CDC, for making decisions with these data. And so once again, the trend would either be positive, negative, or insignificant. And so we wanted to compare the trends across these different indicators. So on top, we have the short-term and sustained trends for wastewater using the influent. And then next was using the solids data from the wastewater. And you can see where in red, that it was at that time period, it would be representing a decrease in concentration. And so the trend would be a decrease. And then in green, this would be a trend that would be an increase. And, that's the opposite. So in red, it would show an increase. And so red is we're concerned. And then green would be showing that there is a decrease. And then we have the cases would be next after the solids, and then the COVID-like illness at the bottom. And just a couple summary points related to this. So if the bar is gray, that means that it was a non-significant trend. So that it was neither increasing nor decreasing. So there were 9 periods of time where the trends across all the indicators agreed. And so, this is 9 periods out of many of these periods, right? And so that was something that was quite noted whenever we think about, do the different indicators provide similar insight on how the concentrations are performing. And, but what was quite nice was that only sustained trends agreed. So ones that were the longer term. I will also note that there were more trends observed, significant increasing or decreasing trends observed for cases. And then it went down from COVID-like illness to solids to influent having the least amount of trends identified. So quickly, I want to just talk a little bit about how the data are used. So as I just mentioned, trends tracking was a dominant way that the data are used, but it was also part of a suite of surveillance tools. So wastewater wasn't just being used in isolation, but that many other pieces of data were being used by public health officials, and wastewater was just one of those tools. And it's also difficult to compare absolute concentrations across the different locations. Usually the concentrations are compared relative to a previous time point at one site. However, you could look at how trends compare across the different locations. And I do want to make note that wastewater is useful for certain things, but not for others. So you can't make individual treatment decisions based on wastewater surveillance tools. And so there's still a need for individualized testing, but you can gain insight on what's going on in the broader population. Further, wastewater surveillance doesn't let you disaggregate your data by subpopulation. However, people have explored doing some of this sub-sewershed surveillance where you could kind of get insight at a neighborhood level. I wanted to show the public dashboard for the state of North Carolina as part of their wastewater monitoring network. There are 21 sites that are being monitored in the state of North Carolina, and they have a really nice dashboard that the public can use to look at trends and to gain insights. One of the key metrics that they're using are looking at a 15-day rate of change, whether or not, how much it's kind of increasing over that 15-day period, and using that as a scale to say whether we're seeing a lot of increases or not. This is another report from the North Carolina Wastewater Network, and you can see that in the report, they still report cases over time, those are the gray bars. And then the wastewater data, you see the green trend line with the green data points, shows the concentrations over time. And then along the bottom is actually this percent change that is also being shown to see over the 15 days whether it's increasing or decreasing. Another metric that has become quite useful to public health officials are looking at, what percentile of current concentrations it is represented in the historical distribution of those concentrations. And so comparing, what a current concentration is relative to all the concentrations that we've seen since we started monitoring. And so this can show, this is across all of the sites, all the 21 sites over time. And you can see in January, you see a lot of red across the sites, and that was the Omicron wave. This is January 2020. And this was the Omicron wave, and then you can see how concentrations have gone down and are starting to rise again. I also want to mention that we are starting to look at variant detection. This can be using targeted gene amplification, so also using digital droplet PCR and just looking for specific gene targets related to a particular variant of concern. And we're also doing some whole genome sequencing of the virus in wastewater to look for genes that are associated with these variants of concern as well. And so looking at using wastewater for variant tracking. In terms of next steps, there's a lot of interest in seeing if there could be other pathogens monitored with wastewater. There are TACMAN array cards that my lab uses where we can scan a sample for multiple pathogen targets in one sample run, and it does offer some quantitative data as well. And so those are looking, and then also multiplex assays. So even with some of the PCR techniques, you can look for 2 or 3 targets in a single run, whereas the TACMAN array cards, you could look up to 40 targets. Metagenomic approaches are also being explored in wastewater and workflows being optimized to look for multiple pathogen targets. So just wrapping up, there are many remaining questions. So just wrapping up, there are many remaining questions to improve how we use these data. Different types of models being used to predict future issues related to hospitalization. So using wastewater to predict hospitalizations. So data regarding shedding rates, the fecal shedding rates of the virus and feces, how travel time in the sewage network impacts what we're seeing in the data. Also incorporating decay rates of the target through the sewage network. Once again, I mentioned the inflow and infiltration and how we're normalizing the concentrations in wastewater and exploring how we could optimize some sub-sewer shed sampling. Wastewater has been a very powerful public health tool in the US, and it's growing in importance as we see less individualized testing, and it can inform testing locations or different messaging that might be happening in an area if we start to see increases. And I know that our public health officials have really started to rely on wastewater surveillance much more recently. And as I said, there's a lot of interest in figuring out how it could even predict things like hospitalizations and mortality. So thanks so much for your time. I just wanted to acknowledge all of the great team that I had working with us, in particular Nadine Kotlars and Francis de los Reyes were other investigators on this work. So thank you so much for your time.
Angela Harris.
Angela Harris, thank you very, very much indeed, my dear friend, for your brilliant presentation. I personally have many, many questions and comments to make to you, but let's move on at this time. We'll hold the questions and we'll make the questions next in the Q&A session. So I remind you all that feel very stimulated by this presentation by Angela, so please make your comments or questions in the chat, and we are monitoring the chat to be able to have this debate next in the Q&A session. I will invite at this time the Dr. Hubert Denise, our dear representative in the UK, the chef of the genomics in the environmental monitoring sector for health protection in the Health Security Agency in the UK, to bring their comments and reflections and tell us a little bit more about his experiences and lessons learned in the UK. Hubert, the floor is yours, my dear friend. Thank you very much indeed for accepting our invitation.
Thanks very much for inviting me, and it's a pleasure to participate to this day. And I would like to perfect the talk from Angela was excellent, and introduce in fact a lot of things that I realize I may not have explained in my talk. So if you could see my slide, I will talk today about the genomic surveillance program that for COVID-19 that we implemented in England. And the idea was to cover, using wastewater, most of the England territory. And this has been done by identifying a number of sites for wastewater collection. And England have been split into regions, like you can see in this part. These regions contain, like other regions, a number of sites who are monitored by sewage treatment work, who are big treatment place who collect wastewater at the end of the network and cover for a large number of people. There's sewage treatment work where monitored about 3 days a week. And then we have also sewer network sites who are grab samples who are collected in cities, generally like this one in Sheffield, example, and they were collected 4 days a week. And between these two collections, we have at least 3 samples per week per area or by site code. And we have, so in total, England monitored 302 sewage treatment work and 187 network sites, covering up to 70% of England population was covered this way. On the map here represents England and the different collection points. On the north, this is Scotland, and on the west, it's Wales, and they do have their own independent wastewater monitoring program. We contribute with them, but the data are not included in the presentation I will give. So we got the samples, and we wanted to use them for wastewater processing. And to that, we use government agencies or environmental agency who take the sample and extract the RNA, concentrate it, and the RNA were then sent for quantification by quantitative PCR, or PCR. Then the remaining sample were sent for sequencing. We use 3 different facilities, one at Liverpool, Exeter, and Nottingham. This sequence was whole genome sequencing, so the whole length of the COVID-19 genome was analyzed by my group using an Amazon Web Service deployed pipeline. We produce then an internal report who are processed by the reporting team in order to provide a report to the local authorities. And in terms of quantitative PCR, we were able to, because we have several points a week, we are able to represent this kind of graph where we have, for example, across, this is across 2-week period. You can see the amount of COVID-19 concentration in terms of GC per liter planted in 1 week. This is another week. And you can see that there is a trend. Some sites are increasing in concentration according to the scale, or some sort of decreasing, and we could represent this at the level of the region, for example, here the northwest. And you can see that for these regions, for example, the regional average, represented in purple line here, was consistently lower than the national average, because represented in a blue line, across the period. This is from the 7th of June 2021 to March, end of March 22. This gap in detection here represents the Christmas period where no sampling took place. And so these regions, you can see the regional average was lower than the national average, and this represents probably a reflection of the northwest area of England, with pretty rural and therefore maybe less contact with people. So this is one aspect, is the confirmation by qPCR. But my group personally was particularly involved in analysis of data for variant detection. And this was done from data sequence at 3 universities. And here I highlighted the detail a bit the data for analysis pipeline. So we use the sequencing files. They are clean and trimmed to remove adapters and read with no low quality. Then aligned to COVID-19 reference using the Arctic pipeline. Then we use 2 different tools. One is called VARSCAN, in order to identify single nucleotide polymorphism, so SNP, or point of mutations in the genome, or insertion and deletions who are shortened in indel signification. Then in parallel, we also use a different tool, which is named COJAP, in order to identify the co-occurrence of several SNPs on the same read. This information from these both 2 tools were fed to an in-house script, auto-screening, who detects the variants of COVID-19 and categorize the detections in terms of confidentiality. No, confident in a score, in a detection. And then an important step we thought for that was we have a manual correction step here, where we take the output of this file and for each variant identify manually assign the status of detection of this variant. For example, in this example, we have a sample who have BA2 confirmed detections, while BA1 and Delta are detected in the sample, but only what we call a possible level. So we think it's, we know it's there, but we don't have the same confidence. And this is done, as I say, by matching the whole sequencing in the whole genome against a known profile for the variants. And having this detection, having done these detections, we were able to represent them across time. And this is a graph represent the equivalent of what was presented before by Angela. So this is from the 1st of May 2021 to the 1st of April 2022. And you could see in wastewater the detections of the different variants done using method just presented. So you can see the delta detection in delta, the detection of AY 4.2, then the rise of omicrons associated with the decrease in delta and AY 4.2, and then the rise of the decrease in omicron BA1 and the increase in omicron BA2. And this corresponds to the Christmas period. Again, no collection took place, and this gap at the beginning of June 2021 corresponds to a logistic issue where not enough samples were collected to be confident in the trend. And using this data, we also were able to look at it on a more larger scale or timeline. And this, for example, represents the detection of from end of January, so end of January 22 to the end of March 22. And you could see that the number of confirmed detections in red or possible detection in yellow decrease across time for B1, while you see an increase of detection of sample across the time. You can see the trend between red to gray, where there is no detection, while in parallel, B2, for example, for the same time period, while in this slide, so the top part is the wastewater detection across time, so the one I presented previously, from the 1st of May to 1st of April, 1st of May 21, 1st of April 22. And at the bottom are the data for clinical detections obtained from the COVID-19 resource provided by the Sanger Center, Welcome to the Sanger Center in England. And you can see that I try to represent put them on the same time scale. You can see that the wave of detection in wastewater are matching the wave detection in clinical cases. This is clinical cases including random testing and targeted testing. And the more, well, we don't present in this slide, but the more sophisticated analysis indicated that the wastewater detections were in sync, or even slightly ahead of clinical detection, exactly what Angela presented nicely before. So the detections were all based, I'll go back to that, to this pipeline, who, as you can see, was able to detect several variants in the same sample. However, we were not able to infer here if most sample was contain B2 or B1. There was no quantification. All we can say is present or not. And so with in collaboration with academic, in this case, Professor Chris Quince from Early Life Institute in UK, we developed an amplicon-based quantification method named CovMix, and the new incarnation of CovMix 2, who aim to be able to indicate the sample if some variant is present in represent 50% of the sample, for example, while other variants represent only 20%. And this was tested against two other published methods, Freya and Callisto, that you may have heard of that, were methods to do the same things across a period of 2 months, from the December 21 to February 22. And well, I will not go in every details of the graph. I'm sorry, I should have maybe zoom in a bit more, but you can see that the pattern from the CovMix to analysis is more closely related to the clinical pattern across the same time period than Freya and Callisto. And this is because CovMix 2, compared to these two methods, have error model who correct for sequencing and PCR error, and therefore give more reliable results. So this is something we are now starting to look at putting in productions. I will give us more insight into the evolution of infection in England. Building on this approach, we are also looking at the de novo variant detections in wastewater samples. Currently, UKHSA genomic method rely on public health England genomic profile for known variants. An example here represented for Delta. Okay, so in the public health England tell us that Delta is characterized by the presence of this list of 13 amino acid changes, and we have the equivalence in terms of genome change and which nucleotide is changed. However, if you want to look at de novo, we can use the fact that we have more than one sample per run or per week in your data. And it's what we have done. So what we have done is to filter out, for example, in the sample, let's say this site have a delta detection, we can filter out the SNP. And because we have 3 days here, we can look at which point of mutations are present across the 3 days in all of them, then filter out the detection of non-variants that we got, and we have residual SNPs that are of interest. And if we do it across a certain several weeks, so 1 week, the second week, etc., we can identify trend across few weeks, who could be indicative or for emergence of a new variant. And some evidence from the rise of Omicron seems to indicate that it is possible to do this way. But also, we are able to identify acquisition of new SNP by non-variant. The virus, let's say Omicron virus, will replicate, and they will introduce our error during the replication, and we could identify this acquisition new SNP. They still will classify the same variant, but they may have new strand. And we have developed this method in-house using historical data, but also with our collaborator in Free Screens as an additional module to CovMix with more sophisticated statistical evaluation. And because we use obviously for COVID, but this allow as well to put wastewater-based epidemiology on the map. And we are now UKHSA now looking at extending the monitoring for other pathogens of interest for public health, poliovirus, influenza virus, norovirus, and enterovirus are currently on the list, but also antimicrobial resistance is considered. And there's different virus, some of them will only involve amplification of particular fragment, some will be qPCR based, and some will require sequencing approaches. So we're looking currently at the best strategy to identify these different pathogens. And of course, we're looking actively how to utilize the multiplexing assay for these different pathogens. One sample given information of different pathogens. And I would like to take the opportunity here to thank different persons in UKHSA, especially my team, the Genômica team, also the research team, the reporting team, the logistic team, and other people that are not included in this slide, as well as collaborators in academia or in other public body, the co-consortium, CIFAS, RM Institute, and the Environment Agency. And I would like to thank you for your attention, and thanks very much.
Uau, Hubert! Muito obrigado, meu caro, né, pela explanação belíssima.
Thank you very much for your amazing explanation. What you have been doing in the UK is amazing. Congratulations. I'm amazed at the amount of sites that you have monitored, and also the interpretation of data, given the different types of virus that you had to deal with throughout all this journey, and the variants as well that have been detected and correlated. I'm impressed. So your presentation has been amazing. Thank you so much for being here with us and for your interest in sharing with us all the work that you have carried out there. Now I will just give a Brazilian touch to this important webinar. And to do that, I will invite Professor César Mota, who is a professor at UFMG. He's also currently the coordinator of the INCT Waste Treatment Plant, Sustainable Wastewater Treatment Plants, and he coordinates also the Wastewater Surveillance Program here in Brazil. César Mota, it's a pleasure to have you here. Thank you so much for having recommended these professionals to join us in this panel today. And now it's your turn. The floor is yours to talk a little bit about the experience of our national network and how we have been dealing with all of that with different methodologies and what we have been doing here in the different capitals of Brazil. The floor is yours then. Thank you.
Thank you very much, Gustavo. Thank you very much, ABES, for inviting me. It's an honor to be here, especially among these two greats that preceded me. Today, let me just start sharing my slides. Gustavo, can you see it well?
Em tela, César. Por gentileza, prossiga.
Okay. So,
yes,
first of all, I'm here, but there are roughly maybe over 50 people, or even more, involved in this work here. So it's a list too big to show all the people involved, but it's a massive effort, and I'm only representing this large group of people and these two organizations that have been responsible for this work here. So INCT, the National Institute of Science and Technology on Sustainable STPs, and also the National Agency for Water and Sanitation. So these are the two major institutions responsible for the work. So thanks the institutions and thanks all the people that have been involved. So as very nicely put by Angela, a little bit of the history, right, of why we are looking at sewage to face this horrible pandemic. So initially, back in late 19 and early 2020, we learned that infected people shed viruses mainly in feces. So we were first very worried, actually. So if people are shedding these, we were worried that there could be fecal oral transmission of COVID besides air droplets and so on. And we did a study, and actually we discussed the possible routes that could happen. And we published this early on, maybe April, I think it was April, maybe May 2020. But what I would like to emphasize is that has not been proved yet. And maybe it won't be proved. Maybe that's never going to be a significant route for transmission of COVID. Until today, we have no studies proving that route. And it's such a relief for us, especially in Brazil and other developing countries that have very poor infrastructure for sewage collection and sewage treatment. With that came actually excitement. So if we can see, if we can measure, if we can monitor the amount of virus particles in sewage, we could actually learn about how the population is doing in terms of the pandemic. And so it came from worry to excitement. So there were some reports early on from the Netherlands and then from Australia, and people were publishing, look, we can detect this virus. This virus is enveloped and it's very sensible to environmental conditions, but they are present in concentrations high enough, and we have the analytical methods to actually measure the concentrations, even though they're very low. So we became very excited and we quickly responded to that. So I like showing this figure here because it shows what kind of role sewage can have in society. So in the late 19th century, we learned, based on the work by John Snow, that water, contaminated water, sewage can be a very significant source of diseases. So that was we learned the main role of sewage in society, especially with very poor sanitation infrastructure. Later on, we learned more about what role sewage could play. So apart from being, of course, it's always going to be a source of disease, but also it can be a source of resources. We can harness energy, we can get nutrients, we can get water for reuse, we can have many valuable things from sewage. And very recently, of course, this is not very new, but it's as Angela also mentioned, it has skyrocketed. The applications of sewage as a monitoring tool for epidemics has skyrocketed in the application. We're only starting to exploit them, to explore them. So it can be a source of disease, it can be a source of resources, but it can also be a very valuable epidemiological tool. And we have much to learn about that. In Brazil, since the beginning, we have been testing very poorly. If we compare the amount of tests we have done in Brazil compared to other countries, we fare very low on the table, lower than some countries in Africa, most countries in South America. So we have been testing very, very poorly, and that's very worrying, especially in the worst times of the pandemic. That was very, very difficult to face. And on in the land of blind, one-eyed can be king. So in this case, the sewage can be our eyes, and we thought this could work. So we monitor the sewage from Belo Horizonte. Belo Horizonte is a very large Brazilian city. It's the greater Belo Horizonte is roughly 5 to 6 million people. And Belo Horizonte has a very centralized sewer system. And most of the sewage that's collected in Belo Horizonte is treated in only 2 wastewater treatment plants, very large wastewater treatment plants. So we monitor these 2 treatment plants, and that's the data we got throughout the pandemic. So we were able to start very early in the pandemic, so April 2020, and the bars show the loads, so the amount of viruses shed, present in the wastewater per day. So the bars show the 2 wastewater treatment plants, the yellow and the blue wastewater treatment plants. The red line shows the suspected cases. What we call suspected cases here is the number of cases of people that show symptoms and looked for help in the health system. And the green line shows confirmed cases. So remember, we tested very poorly, so the data on confirmed cases is very problematic for us here. So if you can see the trends throughout the pandemic from early on all the way until now, we are still monitoring, and we will be monitoring for a while more. So if we can, I would like to highlight also how closely related the data are, especially the suspected cases and the loads of viruses in the wastewater. So we had a very clear first wave here, and the suspected cases show. And I'd like to also draw your attention to the shaded areas. So the very dark shade of gray shows very restricted measures that were imposed here by the mayor in Belo Horizonte to protect people and to control the pandemic. So very dark means shops closed, schools closed, in the strictest of the situations. Lighter gray was less strict, so shops were open and so on, and white or very light gray is even less restricted, so the situation was slightly better. So you can follow the graph slightly better, I think. So there was a very clear first wave, and then we had a very strong second wave that actually lasted for a long time. And it also shows the Omicron wave as well. So very strong also in terms of viral load in the wastewater and also in the number of cases. This next graph here also shows the shaded areas with the restrictions imposed by the mayor. But it also shows the situation in the health system. So here in dark red, we see the number of intensive care beds occupied. The white dots in red here show the number of intensive care beds available for COVID patients. So whenever this line here is very near zero, means collapse or near collapse of the health system. So we reached near collapse or collapse multiple times during the pandemic here in Belo Horizonte, which was not one of the worst hit cities in Brazil by far. So there were cities that were in even worse situations than Belo Horizonte. So this is very sad, and this is very worried, that we got to these points in multiple times, collapse of the health system. The green line shows the beds, the general beds for COVID, so 9 intensive care beds. So again, the full triangles show the occupied beds, and the white triangles show the available beds. So there were at times when there were very, very few beds available, even for regular patients. We have not been able yet to do variant work or variant monitoring or quantification in sewage, but based on clinical variant data, we of course know that this is Omicron, and we also know that this was by far the worst wave that we faced. This was the gamma. This was the gamma wave. And gamma, for international people, gamma was very, very strong here in Brazil. It wasn't as strong. Actually, it was hardly detected in Europe, in the USA, but it was very strong here in Brazil and was the deadliest of the variants here in Brazil. And you can see here the peak for gamma, and also how strong an effect it had in the health system here, shown by the intensive care and the regular beds here. So this was our centralized monitoring, so based on the major treatment plants here in Belo Horizonte. However, like any big city in Brazil, this is a picture of Belo Horizonte. Belo Horizonte is extremely diverse. It's very heterogeneous. So we have neighborhoods in Belo Horizonte that are like Luxembourg or Belgium, or very rich countries with very, very good social and health indicators. But of course, we have the other opposite. We have many neighborhoods in Belo Horizonte that are like many of the worst countries in Africa, where people hardly have water, people don't have houses. So the situation is extremely difficult for these people in a regular situation, let alone especially during a pandemic such as this. So we were very curious to see and to monitor the wastewater in different regions of the city, because of course we hypothesized that the situation was very different from people that lived in the Luxembourgs of Belo Horizonte compared to the Haitis of Belo Horizonte. And we were very interested in looking into that. How? we decided to, of course, monitor both treatment plants, but also we decided to monitor specific regions. So we went up the sewer network. So we collected samples in different neighborhoods, collecting some from the interceptors with the large pipes that we have on the sewer networks. And of course, on purpose, we chose regions of the city with very different indicators of health and social indicators as well. So here are the shaded areas that are monitored by each of the red dots. And this is the table where we use to weight the health vulnerability index that we chose, that we used to choose each of the neighborhoods. So we have very not vulnerable neighborhoods, we have very vulnerable, and we have extremely vulnerable neighborhoods that were monitored. And this is like a flow diagram, a simplified diagram of the monitoring program that we had. So this is the sewer network and the points that we use to monitor. For example, this AWS sampling point represents the sewage from roughly 130,000 people, very actually in the considered vulnerable. This monitors, for example, around 46,000 people, not in very vulnerable, actually not vulnerable situations, and so on. One of the first things that we were worried was if we were looking at different places of the sewer network, we were very worried about the effect of RNA decay in the sewer. So if we were collecting upstream, we were worried, well, if we have a stronger signal here compared to the signal downhill, maybe it's because of decay. Maybe it's not because of the situation in that neighborhood is worse. So that's the first thing we decided to look into. And we used a model to actually estimate the decay of the RNA in the sewers. We of course, we got the samples, actually the information for the sewer network from the local sanitation company. And our results show that the maximum estimated decay of RNA that in our sewage was only 2%. So it was really negligible. It was something that we shouldn't worry about, at least in the context of Belo Horizonte, which is a very hilly city. So because it's very hilly, sewage doesn't take very long from the place where people flush the toilets to the place where we collect the sample, and also to the place where the treatment plant is located. Also, we tried to develop an indicator to actually tell us where or how to identify hotspots within the sewer sheds that we had. So what we did was we decided to develop this IPR, is what we call the RPI, the relative prevalence index. So if we monitor in a sub sewer shed, the sewer here, so we collect the sewer here, and we collect the sewer downhill here in the treatment plant at the end of the sewer shed. We compare the results, the concentrations here with concentrations here. And I will simplify here because I won't have time to explain how we got to this equation, but it was mostly to avoid having to calculate the number of infected people based on the sewer and based on the viral loading feces, because we know that that's not very reliable in terms of modeling, because some infected people shed very, very high amounts of virus, but others don't shed or shed very, very little. So instead of looking to the data that's problematic, we actually avoided that by developing the RPI, the relative prevalence index. That only takes into consideration the concentration that we measure here in the sub-sewer shed and we compare the concentration that we measure here in the sewage treatment plant. So in summary, if the RPI is higher than 1, meaning the concentration in the sewer shed is higher than the concentration measured in the sewer network, if that's higher, we consider that region a hotspot during that period. Of course, it's the dynamic indicator, and it will change throughout the pandemic. And we evaluated the RPI for the first wave. I don't know if you remember the first wave in the graph here, in the previous graph that I showed, was the first wave back in 2020, in June, July 2020, the beginning of the pandemic. So we evaluated the RPI before the wave, the surge, during the surge, and after the surge. And our data shows that the neighborhoods that are shown here in red are the ones are the ones that are most vulnerable. So they have health vulnerability indexes very, very indicated that are very, very vulnerable. So these people, as shown by this graph here, by the RPI, show that they suffer a lot more than the people not vulnerable, which are shown here in green. So the sewer data showed that the situation was very different in different neighborhoods. So people that were in more vulnerable neighborhoods suffered even more during the pandemic compared to the people that were in not very vulnerable neighborhoods. And that's only based on sewer, the graphs that we're showing here. We published this data in this paper here if you want to learn more. So this was the pilot project that I just showed. And the partners were INCT and ANA, the National Agency for Water and Sanitation, decided to expand the project, and as a proof of concept for the national program of wastewater-based epidemiology in Brazil. So the program was expanded to more cities, to big cities. And this graph here shows the locations of all the cities. We have Fortaleza, Recife, both in the northeast of Brazil. We also have Rio de Janeiro, also in the southeast, as well as Belo Horizonte. We also have Brasilia, and we also have Curitiba down in the south. And these are all the partners. And I would like to highlight the importance of all the partners in the sanitation companies. Without the sanitation, the help and partnerships from the sanitation companies, the whole program would not be possible. And I also highlight all the partners in each of the federal universities that are participating in the monitoring. So we please look closely to all the partners that we have, and each of these partners have a large group of students, researchers, and academics involved. We estimate that this larger program that we call the Brazilian Network for COVID Monitoring Sewage, we estimate that the monitored population is roughly 14 million people. Brazil has a population of roughly 200 million, so slightly less than 10% so far. And for each of the cities that we are monitoring since April, and each of the cities, Belo Horizonte, here I'm showing Belo Horizonte, Curitiba, Brasília, Distrito Federal, and Fortaleza. We have the monitoring points for each of them, roughly 10. Some of them have 12 total points. We also have Recife here, and Rio de Janeiro here with all the red points. I'd like to highlight that each of these cities have very different sewer networks. So in terms of coverage, for example, Fortaleza and Recife have fairly poor coverage. I think Recife, both around 40 to 60% of coverage in collection, sewer collection. Whereas Curitiba here has very high coverage, almost 100%, higher than 90% coverage. And also, for example, in Distrito Federal, Brasilia, we also monitored Brasilia, but we also monitored satellite cities. So it was a wider area as well. Other particularities of that are interesting. So Recife monitoring program, because they have a very poor coverage, we were able actually, the whole program decided to actually include some of the channels, the open channels that receive untreated sewage, of course, in rainwater, to monitor. And they have been used throughout the monitoring program for that. So it's both. And also we have a variety. For example, we are monitoring sewage treatment plants. We are monitoring sewage pump stations within the sewer network, and also open channels. These are examples of the graphs that we generate. It's a heat map, and we have the scales. So green when it's not detected, yellow when it's detected but it's below detection limit, orange it's we should start to worry about the concentrations, and red is really high. We should be very worried about the high loads and concentrations. So this is just one example for Brasilia, but we've been producing these graphs weekly for all the cities that are being monitored. We also monitor different points, and we publish these graphs as well for each of the points that we monitor, and we publish the concentrations, so copy numbers per liter of sewage. So, and you can see here throughout the pandemic, this is for Belo Horizonte since April until now, still monitoring. And again, this is the Omicron peak, one sewage treatment plant to the other. We have also been producing these graphs with total loads of virus in the sewage, but also the rates of people vaccinated. So the red line shows the percent of people with the first dose of the COVID vaccine. The green line shows the percentage of people with the second dose, and the purple line shows the number of people fully vaccinated with the third dose, or people with the vaccinated with the vaccines that are one dose only. So you can see that, for example, during the Omicron surge, we already had a high number of people vaccinated with the second dose. We didn't have a very high number of people with the third dose. And I don't know if you remember, but we did not see a very worrying peak of hospital beds occupied during the Omicron surge. And we are positive that the vaccination had a strong effect on that. It's a very good success story here. We have also been monitoring specific points of interest, so points that have very high circulation of people, for example, airports, bus stations, and also elderly shelters. So there are people that are very, very vulnerable to COVID, and we've been monitoring this throughout the program. We have been trying to reach all local health authorities, including the municipal level, also the state level, and also the federal level with the data. And many of them have included many of them have included the sewage data in their weekly discussions, and some of them even publish the sewage data together with the clinical data. So we have monthly bulletins. So we have every month we show all the graphs for the past 4 weeks for each of the partners. We also have special bulletins on specific topics, for example, methodology and so on. We have alerts, for example, warnings. So we publish nationwide warnings when we see a significant increase and sudden increase in loads of virus particles in the sewer. And we also have technical notes and regular webinars. And of course, the idea here is to get the highest number of people informed by sewage data, which doesn't depend on testing, and people can plan better. Not only health authorities, which are very important, but also the regular Joe. We are also interested that we also worry, actually, we make significant efforts to actually get the data to the regular Joe. We think it's important that regular people understand this. Looking ahead, I borrowed this slide from people from the Netherlands, but looking ahead, there are many things yet to be done based on monitoring of sewage. So we list a few of them here, and we are pursuing. So the National Agency for Water and Sanitation, together with the Ministry of Health, we are in talks with INCT, of course, included. We are in talks to create and develop a national network, actually a national program for wastewater-based epidemiology. And it's not going to be focused only on COVID. We think it's important that we look post-COVID into other infectious diseases. There's no shortage of infectious diseases here in Brazil, and also many other areas of interest. For example, antibiotic-resistant genes, antibiotic-resistant microbes as well, polioviruses, arboviruses, and of course variants of concern. So we think there's a bright and long future here in Brazil for WBE. With that, I'd like to thank you all for being here, and thank again all the people involved and the institutions involved and the partners involved, and thank ABES for having us all here. Thank you very much.
Maravilha, meu caro César.
My dear friend César Mota, Professor of the Federal University of Minas Gerais, our partner in the COVID network. César, thank you very much indeed, my dear friend, for bringing all these important information and the work that's been doing here in Brazil. That's a lot of wonderful things that were information that was produced in such a short period of time, and that was very quickly translated. Typically, I like to say that it's applied science. So it's knowledge that left theoretical level and was translated to bulletins and maps and charts and websites, and that can be accessed by the competent authorities, but more than that, by any citizen who is interested in this theme. César, thank you very much once again, my dear friend, for the work that you've done and for representing us within this theme at on this webinar and all the other forums that you have been contributing to on a routine basis over the last few months, notably the work associated to enterology based on that. Well, given the three presentations, the experience of the United States and the UK and the Brazilian experience, I pass the floor now to our dear special guest and General Coordinator of Environmental Health Monitoring in the Ministry of Health, Thais Araújo Cavendish. Thais.
Thais, the chat is full of questions now. And there are a lot of reflections that were shared with us as well. So now, please, let's stir things up and let's make sure that all of our panelists will answer our questions, and let's make sure as well that all of the attendees participate with us. Thais, the floor is yours. You will be moderating this debate, and thank you so much for accepting the invitation to be here with us.
Muito obrigada, Gustavo. É um prazer estar aqui mais essa oportunidade para discutir esse tema de altíssima relevância, que é a vigilância baseada no esgoto, epidemiologia baseada no esgoto, brilhantemente explorada pelos nossos 3 palestrantes aqui presentes hoje, a quem eu agradeço muitíssimo, ao César, ao Hubert, a Ângela, por tanto nos ensinar, né, numa manhã como essa. Eu que, de fato, sou uma aprendiz sobre esse tema, me sinto bastante contemplada com vários novos conhecimentos que até o momento eu não tinha. Então, agradeço por isso, acho que a nossa audiência também sente o mesmo, dado a efervescência que a gente observa no chat, os comentários que têm chegado, diversos interesses sobre diversos aspectos da epidemiologia baseada no esgoto, claro, né, com foco, nesse momento, com foco no monitoramento do SARS-CoV-2, né, para fins de auxílio à vigilância da Covid-19, mas, como muito claramente evidenciado por todos os nossos palestrantes, também já com um horizonte, né, de interesse para que essa mesma metodologia possa ser utilizada no futuro, ou até agora, ou no passado, como já assim foi, para a pesquisa e identificação e auxílio à vigilância de diversos outros patógenos ou substâncias químicas de interesse. Foi bastante útil ouvir os palestrantes nesse sentido, né? Eu coletei algumas perguntas do chat, né? Acho que várias convergem para alguns pontos, e acho até que os palestrantes passaram por várias delas, mas eu vou, antes de começar com a sessão de perguntas, eu vou apenas fazer alguns comentários em relação às 3 falas que acho que, para nós aqui no Brasil, né, e aqui na minha posição como representante do Ministério da Saúde, são pontos que chamam a atenção, são pontos que transmitem para nós, formuladores de políticas, a segurança necessária e os requisitos técnicos necessários para que a gente possa incorporar esse tipo de metodologia, de estratégia, de vigilância na perspectiva de programas nacionais, programas locais, de fato, né? Dado a sua utilidade para a vigilância em saúde e também, claro, para os serviços de assistência. Bom, então, só resgatando alguns desses pontos, né, que eu acho que foram muito relevantes das falas dos 3 palestrantes, a doutora Angela Harris, né, ela já, acho que eu vou utilizar sempre essa pirâmide que ela trouxe logo no começo da fala dela, né, onde a gente consegue enxergar claramente, a gente consegue enxergar claramente a distância entre a informação que temos, todas as pessoas que eventualmente estão passando por algum tipo de infecção, estão doentes, passando pelos testados, né, os sintomáticos, dos sintomáticos para os testados, dos testados para os notificados, e assim vamos até essa pirâmide numérica chegar aos indicadores que nós todos pretendemos evitar, que são os óbitos. E por que não, né, pensar em evitar também a hospitalização, que é outro degrau que aparece ali naquela pirâmide. Esse tipo de linguagem deixa muito claro para o formulador de políticas em saúde a importância de metodologias desta natureza e todo tipo de estratégia auxiliar para a vigilância epidemiológica quando estamos falando de epidemias, ou no caso, né, pandemias que realmente assolam a saúde da nossa população, né? E a gente viu na fala dos 3 palestrantes, né, isso apareceu bastante claramente, a relevância e a facilidade de usar a metodologia baseada no esgoto. Quando a nossa população tem acesso, né, é conectada à rede de esgotos do município, da região onde vive, isso facilita todo o trabalho, né, de pesquisa e de captura, monitoramento de vírus nessas circunstâncias e, portanto, retorna, né, o esgoto, o abastecimento de esgoto na população pode ser utilizado e retorna para a própria sociedade como uma ferramenta, né, como uma fonte de informação riquíssima sobre o seu estado de saúde. Então, a gente, com esse tipo de trabalho, ainda reforça mais um pouco a importância da cobertura dos serviços de saneamento em qualquer país. A gente viu a diferença, né, na fala dos 3 palestrantes, a gente viu a diferença do que uma cobertura de esgoto e saneamento pode favorecer, né, a população quanto a esta metodologia em específico, e quanto isso pode ser devidamente incorporado, né, na política pública. Então, várias das perguntas têm a ver com isso, e acho que isso é um ponto para a gente ressaltar como a constante relevância que a gente precisa sempre lembrar aqui no Brasil, constante relevância e necessidade de que a expansão dos serviços de saneamento aconteça, né, para a população brasileira. A gente ouviu também, né, na fala da professora Ângela, toda uma necessidade de que a metodologia conhecida até então, antes da pandemia, fosse devidamente adaptada, né, e incorporada à necessidade de monitoramento do novo vírus, né, do SARS-CoV-2. E isso, de fato, para nós, né, que somos formuladores de política, é o tipo de processo que vocês na pesquisa vivenciam e que a gente sequer imagina, né? Tem todo um trabalho de adaptação dessa metodologia. Então, isso eu também já me direciona algumas das perguntas que vieram no chat, do quanto esse trabalho cuidadoso de se entender se todos os métodos empregados para outros vírus, outros patógenos, se adequa, né, de maneira fidedigna a novas necessidades. Então, isso ficou claro na fala dos 3 palestrantes, e isso é algo que eu gostaria de ressaltar do ponto de vista do rigor técnico-científico que a gente assistiu nas palestras de vocês, né? Aqui, eu aprendi muitas coisas na palestra da doutora Ângela sobre a utilização de sólidos ou de águas residuais, e isso realmente é algo novo para mim, então foi muito interessante entender esse percurso metodológico que foi feito pela equipe, né, na Universidade da Carolina do Norte. E eu finalizaria meus comentários sobre a palestra da doutora Ângela mencionando que a forma como esses dados foram publicizados, né, a forma como esses dados foram entregues à sociedade dos Estados Unidos, né, isso de fato é um objetivo que nós aqui no Brasil buscamos, né, a fala do Dr. César ao final da nossa sessão, evidenciou isso, né, o quanto nós estamos buscando com as nossas equipes de pesquisadores uma forma de que a gente possa traduzir esses resultados de maneira bastante clara para toda a população brasileira atendida, né, por essa vigilância, e que a gente possa potencializar esse trabalho, melhorar a nossa forma de comunicar isso de maneira bastante uniformizada, né, e compreensível para todos os que têm interesse, né, nesse tipo de informação. Bom, eu passo então a breves comentários da palestra do Dr. Hubert, que me surpreendeu bastante, né, ao ver o mapa da Inglaterra totalmente coberto, né, com 74% da população inglesa coberta, né, com um monitoramento e com uma rede de esgoto bastante satisfatória do ponto de vista, né, comparativo, quando a gente pensa países como o Brasil e outros, né, e isso é o tipo de coisa que brilha aos nossos olhos, né? É um tipo de futuro que a gente imagina com a evolução de políticas de saneamento, né, em todo o país que ainda precisa fazer, né? É interessante ver na palestra do Dr. Hubert o quanto isso foi dado destaque, né? O quanto a metodologia empregada foi capaz de diferenciar as variantes, né, do novo vírus. Então, isso foi fundamental do ponto de vista da estratégia de vigilância genômica genômica, né, aqui no Brasil, conhecer aqui, né, no ponto de vista da vigilância epidemiológica, conhecer as tipagens dos vírus, entender quais são os seus potenciais de virulência, quais são as suas características do ponto de vista do agravamento da infecção. É muito interessante que isso também seja possível de observar no monitoramento baseado no esgoto, e o que nos evidencia a relevância e a validade dessa metodologia, inclusive quando a gente pensa em diferentes variantes, né? Uma clareza que a gente tem em todas as falas, também aparece na do Dr. Hubert, é o quanto as detecções virais, a carga viral identificada, ela está sempre em sintonia ou um pouco antecipada em relação... Desculpe, acho que eu preciso mudar meu canal.
I'm sorry, I need to change my channel here.
Desculpe, pessoal, eu estava no canal...
I'm sorry, I was in the untranslated channels. I went back to Portuguese now, if there was any failure here. Okay, thank you. Well, so it's important to note how much these detections were aligned with the clinical indicators of clinical testing, but also how much they were even ahead of the clinical indicators that were observed in all of the epidemiological scenarios of the countries we mentioned here. And of course, Dr. Hubert reinforces the theme that it is not just on a national level, but it's important that all researchers and policymakers see the potential in this methodology for other types of pathogens which are of interest. And I would highlight for the existence of microbes, which is a possible potential interesting use for the use of this methodology from the point of view of government policy, since this seems to be one of the major challenges from the point of view of the policies for the use and improvement of research for CISCOT and the solid waste treatment policies in Brazil, and how much this impacts our lives. And finally, before moving on to the questions, I will just make some brief comments about the presentation of Dr. César Mota, who is an old friend and partner. It's difficult to say old, but we spent some good many years together, working together, and we know about each other's work. And it's very, very gratifying for me as the representative of the Ministry of Health of Brazil to count on a team like the INCTs and Sustentáveis and all the framework of universities associated, partnered with the project, the National Water Agency, and how happy we are and how much we can trust the work that is done by this whole team. That, as César mentioned, it is an enormous team which supports the work that was presented here. And the César's presentation, I think, brings an important focal point that appeared in our chat about how much in the Brazilian scenario how we have important vulnerabilities within our territory, all of them connected to our policies and lack of policies for housing, sanitation, urban planning, and how much this has an impact on the potential use of this methodology. So I think there's many questions that were raised that were answered in César's presentation, and I thought it was very impactful how much just with the data obtained from this methodology, how they can show the discrepancy between the aggressiveness and the viral load of a pandemic like this one in neighborhoods and among populations that are highly vulnerable in comparison to populations with less or almost no vulnerabilities at all. And this is a landmark finding, and it's important that Brazilian public healthcare policies are aware of that. And I speak from the Ministry of Health that it's important that we here in the Brazilian unified healthcare system understand that this type of monitoring and surveillance has an even greater value to add to these populations, given that their access to all health services we know there's much less. And this is part of the vulnerability indicator that they have. And so I was very impressed with this data, and I think we need to translate that into actual impactful actions. And with this, I close my comments about Dr. César's presentation, indicating that in the case of Brazil, all this team working on this scene have, without the shadow of a doubt, have been having very important conversations with local healthcare authorities and with the healthcare authorities here in the Ministry of Health. We've been able to work very, very well on a local level, especially. We have efforts ongoing here with the allocation of resources from the central public health laboratories to collaborate with this type of methodology throughout Brazil and help local authorities in this process, and to be at the service of the sanitation service to discuss the results. And we have worked here internally as well along the lines of internalizing the program for a surveillance program based on sewage monitoring, and thinking of all the other ways we could use this type of methodology. So I could speak on this ad infinitum, but as we don't have much time left, I will invite you to have a look at the questions you've received in the chat, and I will do that here in the file I prepared. And I would like to begin with the question from Maria Angelica Iacuna. I think I will post this question to Dr. Angela Harris, who was, I think, the person who mentioned this in a little more depth in her presentation. And Doctor, please feel free to respond. Of course, if any of the other speakers want to chime in, please feel free to do so. Maria Angelica Iacona asks, the PCR test did test positive for many different pathogens. Isn't there another safer way to detect the COVID virus in the sewage systems or wastewater system? Dr. Angela, I hope you understood the question, and please, if you would care to comment on it.
So I think I understand. So the predominant methodology for detecting SARS-CoV-2, both in clinical samples and in wastewater, is using a polymerase chain reaction, a reverse transcriptase polymerase reaction, because SARS-CoV-2 is an RNA virus. And so for clinical testing, so if you do these PCR tests, they will use the same gene targets often as we are using in the wastewater. And so there are multiple different gene targets that are sensitive and specific for SARS-CoV-2. In the US, we are using the N1 and N2 gene targets. So we're actually using two targets, and we see very strong correlation between those two targets. And we're using digital droplet PCR in my lab because this technique has been shown to handle inhibition a little better than quantitative PCR, qPCR using standard curves and qPCR reaction mixtures. And also digital droplet PCR is a little more sensitive. So you can detect often lower concentrations of the viral RNA in your sample than the qPCR technique. Some people are using some of the are interested in exploring using like metagenomic approaches where you do, more non-targeted sequencing to understand the microbial population. However, if your target is very rare or in very low concentration, some of the metagenomic approaches can be quite challenging.
Perfeito, doutora Angela. Agradeço muito.
Perfect, Dr. Angela. Thank you so much for the clarification. And with this brings us to the next question, which I identified here. It is a question that is not have the questioner did not leave their name. And this is a question I would direct to Dr. Angela and Dr. Hubert. What is the size of the team for the work to collect samples in the UK and the United States? If the question is not clear, please let me know, I'll repeat it.
Yeah, so I can just briefly comment. So there was a quite large team early on when we were trying to validate different methods. And so there were 4 different labs involved in kind of comparing different techniques. And we were also interested in kind of inter-lab comparisons. So like my lab would process the same sample as another lab, and we would see how consistent the findings are, because there is variability kind of in every processing technique. And it's helpful to quantify that variability so you understand kind of the uncertainty around these measurements. In terms of the US, so at the wastewater treatment plants, there are staff that collect the samples. And then for North Carolina, we either have the samples shipped or we have couriers that bring the samples to the lab. And then in the lab, we use a combination of technicians and kind of automated robots in the lab to help with the sample processing. And so, I think we're able to do quite a few number of samples within a short amount of time. So for instance, we process around 36 samples that we can do extraction digital droplet PCR within a 48-hour period if we kind of have enough staff. Sometimes it takes us 72 hours to go from data collection to data processed and reporting. And that's a team of 3 technicians that are working on that.
So for us,
Professor Hubert, we can't hear you.
Because we talk to the Environmental Agency or not even
Professor, Professor Hubert, você pode repetir a sua
Professor Hubert, could you please repeat your response, please? We weren't able to get your audio.
Professor, temos algum problema com a sua conexão, que a sua...
Professor, I believe we're having some issues with your connection because your audio is coming through very chopped up. Would you allow me to pass on to the next question? If the connection improves, we can go back to this question, please.
Is it better?
No, Professor, unfortunately, it's not.
Pessoal da tradução, vocês conseguem ouvi-lo melhor? Eu não... Não, né? Ok. Professor Hubert, está um pouco difícil capturar o seu áudio.
Professor Hubert, unfortunately, it's very difficult to hear your audio. Please check if you're having some connection issues that can be resolved, and we will be back in brief shortly to hear your response. In the meantime, we will move on to other questions. Thank you for your understanding. I will now pass to a question to Professor César that came from Denise Tatiana da Costa Santos. Professor César, I'll take advantage of this opportunity to ask 2 questions at once for you because the number of questions that arrived for you was very high. So I will try to summarize at least on 2, and I will ask the rest. The first question is from Denise. Professor says that the use of sediments as samples, was that used in a study in Brazil? Or if not, are you thinking of applying this? What is your opinion about this sampling for the Brazilian reality? And the other question that I will add into that, because it came from many different, it was expressed in many different ways from André Vivaldo Silva, is about the participation of states of the northern region and northeast region in the monitoring network, about this process of configuring the network and how the decision was made and why there isn't such a massive participation of other states in the north and northeast region. That's it, Professor César. Please feel free to respond. Thank you, Thais. Some questions here from Brazilians. I will answer them in Portuguese. So, the first question about the solids. There have been some articles that prove a very good correlation between the loads found in the solids, in the primary solids as we call them, because in the sewage treatment there's many solids that are generated, but I won't get into too much detail about that because maybe everyone in the audience won't be able to understand. The primary solids are created in the primary sediment section. These are very common in the United States and in the UK because they use the technology of activated solids. So we have a primary sediment generator in the beginning of the process. This virus of SARS-CoV has a very great affinity for solids because of its viral load and for other reasons. So it very easily is absorbed and it adheres to the solids, in the case of the solid sewage. So the researchers in the UK and the US can see that there is a very strong correlation between the clinical samples of confirmed cases with the loads found in the sludge. And Professor Angela Harris also showed the comparison of the influent and the sludge. So there's information both phases, the solid and liquid. Here in Brazil, there are very few examples of primary sediment separator in the, we use various other technologies other than activated sludge. So we have lagoons and various other, there's very great diversity of technologies used here. And not necessarily do they use the primary sludge remover in the beginning. So for us, there was not that much interest in one of the major treatment stations here has a primary sediment separator, but we're already monitoring the liquid phase, and we continue to monitor the liquid phase because we know that we have valuable information in the liquid phase as well if we need to find the sludge. So the information, I'll answer the second question now. The network, the structure in the network was especially based on the capillary of the existing networks. CTTE was already formed, and so this rapid response that we were able to obtain together with the ANA, the National Water Agency, to put together this program, monitoring program, was because of the team that was already put together. The laboratories were already put together, the people already involved, so we were able to rapidly respond. And we already had these partners in place who are they will take the initiative. So it's in Recife, in Fortaleza, and Rio de Janeiro. So these were already members of our network, and we added some new and important partners in the case of the COVID network, which were the Federal District and Curitiba. Curitiba was already part of another network, as we mentioned. So this was a pilot project. It was an initial project, and we tried to include more cities with different situations, different realities, like a proof of concept, if you will. But I hope the national program that is being developed will be much broader and include cities from different sizes, medium, large, small, in all regions of Brazil. And I think that this would be the most appropriate way to validate the program, if possible. Wonderful, Cesar. I thank you very much for your comments, because in point of fact, this is the potential response we have for a situation that has become very urgent, like in the case of the pandemic. And you're right to speak to the future in that regard of meeting the needs of regions that need this type of service within a program. Thank you very much. I will take advantage of this space between one question and the other. I will pose the last question to you all to read the response of Dr. Hubert, who was not able to tell us with his own words, but he wrote it here in the chat. So I will read his response so that our translators can translate a little better than the audio. What he responded to us about the size of his team that is needed in the process, he says this.
Organizing the logistics. In time of analysis, my team, 3 people, including myself, they process about 1,500 samples per week in about 48 hours. Isso. Então, muito obrigada, Dr. Hubert,
So thank you very much, Dr. Hubert, for that response. And I will then move to our final question. Unfortunately, we have a great many questions, but time will not permit us to ask them all. So the question is to all 3 speakers, which I think everybody touched on this in some way, but I think it's worthwhile to us to close these final reflections by each of you. The question from Vanya Vicente, and she asked if these results are being adopted or were adopted for lockdown measures. And I think it's good to expand on Vanya's question. So any control issues, not just lockdown issues, but any pandemic control measures in each country where these studies were undertaken. So I will close with Vanya's question exactly to take advantage of your presence to understand a little better how was this experience, how is this experience of dialoguing about this data with the local and national healthcare authorities. We'll start with César, and then we'll move to Dr. Angela. Dr. Hubert, unfortunately, continues to have connection issues, and we will not be able to hear his response at this time, but I hope he can send us this in the chat. Dr. César, if you please. This is a very important aspect. How are we using this? This is a concern that we all have. And it is not an easy thing to answer. We haven't solved this problem yet. I think we have a long way to go yet. But we know, how do we know it? We know that we made small surveys with healthcare authorities here in Belo Horizonte and in the state of Minas Gerais, and among all our different partners as well, that have reported to us how they are using this data. So we have examples within our network of municipal authorities who have used the sewage data as an indicator for the bulletins of the local government together with the clinical data. So this is the case that was happening in Fortaleza. Here in Belo Horizonte, we have a confirmation that this data generated by us in the sewage is part of the agenda and is in part of the discussion, the weekly discussions of the municipal COVID response committee, and also on the state level. We have confirmations that the authorities that manage the international airport here in Confins also include this data in their discussions every week that they as soon as they receive them, and they take it into account. And also, whenever we detect any positive sample in the homes for the elderly or senior living facilities, we list them. And as soon as we are certain of it, we send an email notifying with that notification to the municipality that is responsible for these centers, and they have used this data to help prevent cases among these senior citizens. So we have concrete examples. We can have more, of course, but we have examples of direct actual use of this data. So the mass media, I think some people have noticed since the beginning, this data from sewage was discussed on major nightly news programs and weekly news specials and social media. All possible media it has showed up. So we have confirmation that this data has arrived at in the hands of regular people, regular citizens as well. This is just the start. We have a long way to go yet, but the potential is definitively proven, in my opinion. Perfect, César. Thank you so much. I fully agree with your point of view. speaking for Brazil. So we will continue to work so that gets better. I think this is the missing link to make the connection as far as government policy. Now we pass the floor to Dr. Angela to close our Q&A session with this response.
Great, thank you for the question. So early in the pandemic, whenever we were beginning to look at wastewater surveillance, we actually asked health officials, what type of proof do you need? What type of data and analyses do you need in order for you to feel comfortable making decisions with wastewater data? Because, you know, people are used to indicators that they're familiar with and that have kind of a historical record of being indicators that are robust enough to make big public health decisions. And so there was a lot of working early on to identify, you know, relationships with the clinical cases, but recognizing that even the clinical case data, right, has limitations, these points around testing penetration and asymptomatic individuals. And so realizing that, is there really a gold standard that we have? And I think the UK is like a great example where they had so much clinical testing, you know, I think relative even to places in the US. And so we worked very early on closely with understanding the types of analyses that need to be done and the evidence that would need to be presented in order for people that are making these decisions to use these types of data. And so once we had some of this evidence of the strong relationships between the wastewater and the more conventional indicators, public health officials, you know, people in our Department of Public Health really championed the idea and the use of wastewater surveillance. And so they have taken a leadership role working with the Center for Disease Control at a national level as well, and learning from other public health departments on how they are using the data and how the data are being presented to the public. That, you know, it's really this kind of leadership and championing done from public health officials, and us working closely with them to like help make sense of the science so that it can then be used. And I would say, you know, it's only recently that the wastewater surveillance is really kind of playing quite a key role, given that there has been such a decrease in testing that's occurring. But I think always it will be one of many data points that are used to make these types of decisions. You know, there's a lot that these are complex decisions that are made in terms of, you know, stay-at-home orders, you know, closing certain things down, mask mandates, right? These are very political decisions as well. And so I think it's just recognizing that more data to make better decisions is helpful, and that wastewater is definitely a piece of that. But I also want to mention that a lot of this leadership has been happening at the state level through conversations with a national organization with the CDC, and at the county level, they have been, you know, learning a lot from what the state is doing and how the state is using the data. And so it has taken the leadership of the state to help support some of the decisions and how they're using the data at the county level. So that is another piece of it, that there's a lot of learning that happens across these different organizations.
Excelente, doutora Ângela. Aqui temos uma realidade muito similar.
Excellent, Dr. Angela. The reality is very similar here. It's excellent to hear about this point of view from a country like the United States, in which the territorial sizes are similar to that of Brazil, and which the arrangements, interfederative arrangements, are very complex. So it's very good for us to be able to learn a little bit more about the experience of articulating these results in other countries. Thank you very much. I would like to inform everybody that Dr. Hubert will not be able to continue with us. Unfortunately, he had connection issues that proved irresolvable. So he will try to answer everyone's questions by email when possible. And once again, I thank all our speakers, Dr. César, Dr. Angela, Dr. Hubert, and also our coordinator for the brilliant moderation of this panel. And I'll pass the floor back to him to close today's activities. Thais, my dear friend, thank you very much indeed for your moderating, for this technical dialogue, and we have seen the work here at a very close level that you've done with the Ministry of Health. And we are very proud to know that we have very important civil servants taking the lead in challenges, in challenging conditions, but with extreme competence to produce excellent results. So thank you very much, Thais, for taking the lead on this. Thank you very much to all our guests who've been with us in this special session of Brazil Water Week. Dr. Angela Harris, directly from the United States, Dr. Hubert, Denise, from the United Kingdom, and Dr. César Mota from Brazil in Minas Gerais, specifically bringing their Brazilian contributions regarding sewage monitoring that has helped us a lot in facing COVID-19. My dear friends, this video will be available on the YouTube channel of ABES. So if you want to watch it again, or if you want to share it with somebody, please feel free to do so. I might add on the ABES website, www.abes-bin.org.br. abes.bin.org.br access to the complete program and agenda of the Brazil Water Week special session. So the link for the agenda is being informed in the chat. Please feel free to access it. And stay tuned on our program and participate in the next events. There are very, very interesting subjects and very important subjects being talked about. And they will be broadly talked about here in the Brazil Water Week. Tomorrow, 10 a.m. Brazil time, we will have one more special session of the Brazil Water Week. And the topic to be addressed will be the innovative mechanisms for financing and credit for reaching the SDGs, the Sustainable Development Goals. My dear friends, our great event of the Brazilian Water Week is just around the corner. It's the most important international event on water and sanitation held here in Brazil, and it will be held between the 27th and the 23rd and 27th of May of this month. So in the next few days, and through a exclusive, interactive, completely online platform. You will have access to more than 110 presentations from Brazil and 19 other countries. So we will have more than 30 hours of content. And beyond the content, the ordinary content, we will have special sessions and content that will be shared in the intervals between the talks. This morning, we recorded with Professor Dr. Indiana from Estadual from India, who bring important information about the work being done there, and for the Brazil Water Week connection. Well, access the website that is on the chat to get to know the topics and to sign up for this event. You don't want to miss it. One last message. Over the next few months, on the 29th of June to the 1st of July, we will have the Portugal-Brazil Symposium of Environmental Engineering, the topic of water and sustainability, challenges and actions, also available through the website on the chat. So just read very carefully the content of the 20th symposium. So join ABES, strengthen your position. If you want to join ABES, access our website, click on the link that's in the chat, and of course, participate in this movement to create connections. As you see in the chat, we've had a meeting here of the professionals that was educated by a professor who is also here monitoring it. So the ABES Connect has been a great platform for integration of knowledge and interest. Sector. So that's it. Thank you all. Thank you very much to everyone who stayed with us throughout these last few hours. And I would like to say that this webinar, in my opinion, brought content that is extremely important, and this truly thought-provoking, and I think that science is ready, willing, and able to help us face these challenges we face on a daily basis. Stay tuned, and we will see you again soon in the Brazil Water Week. Warm greetings to everyone, and we will stay in touch.
Here we go.